Did I hand it out the other day? Oh, did I just give it to you yesterday? I'll give it to you.
What?
The homework that's due tomorrow?
Wait, can I have another one of
With
those?
that whole package?
Yeah. All right, so the topics we've left to cover here in genetics are a little bit about population genetics, and a little bit about modern genetic engineering, biotechnology, things like that. So this term population genetics refers to the study of the genes present in an entire population of organisms. Okay? We can look at a population as a whole and try to study the patterns of the various genes that are contained within that population. And that's what population genetics is, sort of a branch of genetics that people study. And so it's the study of these frequencies of various genes within the population. Now you need to know some terms here. So if you think back to ecology, when we studied in 7th grade, a population is a single species in a certain location. So we're talking about the genetics of a population. Obviously they have to be connected in some way. Okay? You're not going to study two populations separated by thousands of miles. You're going to look at one specific population, one specific area, and then you could look at which genes are present in that population. How common are they? We call that the gene pool. So when we look at a population, we look at certain genes in that population, we call that the gene pool. And the more frequently you find a certain allele, that's its gene frequency. Okay, so for example, if we were to imagine that this class is a population, we could look at the frequency of the gene for having 6 fingers. And we would find that it's pretty low. It has very low gene frequencies in our class. That's an example of how you would study it. And so in the early 20th century, some researchers started to devise a mathematical model that can predict the frequency of various genes in a population. And the scientists were named Hardy and Weinberg, and they came up with a principle that in certain populations, the frequency of the genes tends to remain stable. That the frequency of having certain genes is stable if several criteria are met. And Hardy-Weinberg equilibrium is a topic that may be on the SAT. Will not be on the readings exam. And so the conditions that are necessary in order for this equation to work, in order for it to accurately predict the gene frequencies, has to be a large population. Okay? It doesn't work in small populations. Individuals need to mate randomly, and there is no selective pressure. There's no migration of organisms into or out of that population. And there are no mutations. If all of those conditions are met, then this equation can predict the frequency of various gene combinations. But how often do you think those conditions are met in an actual population? Rarely. Almost never. Okay? But that's okay. Because what we can do is scientists use this Hardy-Weinberg equation to predict the frequency of certain genes. And then they look at an actual population. When they find that an actual population isn't meeting what they would predict through this Hardy-Weinberg equilibrium equation, they can then look at, okay, well, why not? Is there selective pressure in terms of mating? Are organisms choosing a mate based on certain characteristics? Is there a lot of migration into that population. It could allow them to sort of look at the reasons of why this Hardy-Weinberg equilibrium is not in place. The way the mathematical formula works looks a little bit complicated. Okay, the Hardy-Weinberg equation is p squared plus 2pq plus q squared equals 1.
Is that on a reference table?
No. Usually they would give it to you.
Okay.
It's I'm not a math teacher. It's sum of squares, is that what it is?
Parentheses p plus q squared.
Yeah, it's p plus q squared, right? Yeah. All right. Thanks. So what does this equation actually mean? Well, you have to know some things. So you have to know how the gene frequency of the dominant allele. Okay. And that's what p is. It's a decimal. So if the dominant allele is found 75% of the time, that's the gene frequency, p is 0.75. Q is the frequency of the recessive allele. So obviously, what would it have to be? If this is just two alleles, the dominant is 0.75, the recessive would be 0.25. So what do you think, if you don't look, what do you think p squared represents? Yeah, like I said, if you don't look, ask me actually think. But yeah, it's the frequency of finding a homozygous dominant individual. 2pq, when you do the math and you multiply the numbers, that would be the expected frequency of a heterozygous combination. And q squared is the frequency of a homozygous recessive.
So squared is homozygous and
Yeah,
times
dominant homozygous, recessive homozygous, and heterozygous. So that's what the numbers show.
So P and Q is for how much the genotype shows.
So P and Q are the frequency of the individual allele. How often is
So
the
how much
dominant
shows up in
allele?
the genotype of it? Like how
Yeah, in the whole population, yeah. How frequent is it? How common is it? And then you could do the equation, you could predict, okay, this is what we expect. We expect to find, 33% homozygous dominant individuals, and so forth. All right. The most common question, though, when you get to Hardy-Weinberg is that why is it not usually realized in real life? And again, it's because these factors are not met. And obviously that should make sense now, that it's not going to meet these predicted values if we have new genes coming into our population because of migration. Then that's going to introduce new genes that we didn't account for in that formula. Okay? If organisms aren't meeting randomly and choosing based on those genes, that alters it as well. All right. The last section in our notes is about modern genetic technology, sometimes called biotechnology, biotech. This is a huge area of study. And when you get to high school, you could take biotechnology. It's take it next year. And so in biotechnology is sort of manipulating organisms, analyzing using modern tools the genetics of certain organisms. It's a wide field, and there's lots of things you could do. When we say genetic engineering, that's not really a great term, but that's what we're going to use, is altering the actual genetic code of an organism. We can now take a gene out of one species, out of the DNA molecule of one species, we can cut out a gene and basically paste it into the DNA of another species. Okay? We can sort of cut and paste genes from species to species. And one of the reasons we can do that is because the genetic code is what? It's a term we use. Why can we take a gene out of a bacteria, put it in a human, and it will still work? Genetic code.
So genes are made of the same stuff.
Yeah, it's universal is what we probably say. That all DNA is A's, T's, G's, and C's. And the codes, remember the codons, every 3 bases is a code for one amino acid, and that's a universal code. whether it's for bacteria or a fungus or a human or a tree, the code is the same. So those genes are transferable. And this has been done for about 30 to 40 years. 1970s, they were the first transgenic organisms were produced. That's an organism that has genes from another species in it. And so the simplified steps, we're not getting into all the details. First you need, so obviously the genes are stored in DNA. So you need to extract the DNA from an organism. Okay. You know, it's done, use chemicals to sort of break up the cell membrane. You use various tools to isolate the DNA. Okay. You can use tools to then amplify the DNA and allow it to copy itself many times, so you have a lot of it. Okay. And usually always in genetics, you're trying to remove one specific gene. So if I want to take the gene, and the example we'll talk about, and probably the one you'll see on tests and stuff, is human insulin. So within our chromosomes, in our genetic code, we have the recipe for how to build the insulin hormone. Okay. It's in the DNA. We want to remove that, sort of cut it out. We can then take that gene and put it into another organism. And so the thing that we do, there are enzymes that do this. Enzymes called restriction enzymes can cut the DNA in specific places. Part of the lab that you guys are going to do, you won't do that section today, but the next slide, is you're going to be simulating this, and you're going to use scissors to cut out strips of paper with DNA based on it, but that's what a restriction enzyme does. It cuts between very specific bases. So a restriction enzyme will recognize a sequence like AA, TT, GC, and it will always cut that DNA molecule between the G and the C. And there are thousands of these restriction enzymes that scientists have isolated, and so if you know which restriction enzyme you want to use to remove a certain gene. So basically you extract the DNA and you cut out the gene of interest using a restriction enzyme. You then take that gene, and often it's put into a bacteria. Bacteria have unique circular DNA, it's called a plasmid. Okay? And you cut the plasmid open, it's a circle, you cut the plasmid open, you put your gene in there, and basically close it back up. And now you have this circular strand of DNA, but included is now the gene that you just pasted in there. In the case we're talking about, the gene for human insulin. You can then put that plasmid back into a bacteria. And now that bacteria is a transgenic organism. It has not only its own bacterial DNA, but it actually has a human gene in it, because that's what you put in there. And so if you grow that bacteria in a culture, not only will it produce its own proteins from its own DNA, but its own protein synthesis structures are going to also make the human protein as well, okay? Because it has that gene. We say that bacteria has been transformed. And that's where human insulin comes from today. Used to be people that were diabetic would have to take pig insulin. That's where they would extract insulin. And that's what they would inject to help control their blood sugar. Today, modern insulin comes from bacteria. Scientists did this exact process. They took the gene for human insulin, put it into bacteria, culture those bacteria, so there's millions of them, allow them to grow in huge vats, and then they extract the insulin, because those bacteria produce human insulin. It's identical to the insulin you would produce in your own body, because it's working off of that gene that we put into the bacteria. And there's lots of variation to this. You can take lots of different genes. They don't have to be put in bacteria. They can put in other organisms. And this is what's used to produce transgenic crops, for example. So we have many, and it's an area of controversy today. Have you heard of GMO foods? What does it mean? Organisms. So some people are very concerned about genetically modified organisms. That we shouldn't be taking genes from different species and mixing them together. People are worried about the health consequences of that. Some people currently are arguing that all foods need to be labeled, whether they are GMO, they contain genetically modified organisms as their ingredients. Okay. Other people on the other side would argue, well, that's not necessary. There's never been any research really showing that these GMO foods are dangerous. There's no data to say that we need to scare people by labeling these things. So it's a back and forth sort of argument. But some uses, insulin, for example. Some types of corn have been genetically modified to be resistant to herbicides like Roundup. You know what Roundup is? You may have it in your garage. Spray it on a plant, kills plants. Kills almost all plants. You spray it on weeds in your sidewalk and they die. Well, scientists have taken, or not scientists, well, yeah, scientists working in corporations, think Monsanto, developed Roundup Ready corn. And they took a gene, some plants happen to be resistant to Roundup, it didn't kill them. They took the genes responsible for that out of that plant, put them in corn, grew the corn, and now we have corn growing that's not affected by Roundup. So people probably say, well, why would you do that? Now then, farmers can take and spray Roundup across all of their fields. It will kill obviously any weeds and plants that shouldn't be there, but because the corn are resistant, they're not affected. So they aren't affected by it. It's a way of killing off the competition for the corn. They've put genes, some plants produce their own insecticides that keep insects away from them. People have taken those genes, put them into plants, so that plants produce their own herbicides that are not preyed on by other insects. Golden rice is a type of rice that's been genetically modified to be more productive, to provide more vitamin A, that has helped millions of people get the nutrients they need in poor countries. Plants have been made drought resistant by giving them certain genes. So there's lots of ways that this is used. They can take genes to produce certain antibodies, put them into cows, and have them produce these antibodies in their milk. They can take basically the genes that produce proteins that they would put in a vaccine, put them into plants, and they can grow potatoes that provide the same function as getting a vaccination shot. So there's lots of, when you start mixing and matching genes from different species, there's all sorts of different things that you can do. We have gene sequencing equipment. Used to be, it's very difficult to figure out, to take a sample of DNA, figure out the A's, T's, G's, and C's that were in it. It was very labor intensive. In fact, 15 years ago or so, there was the Human Genome Project, which was this big international project to try to sequence an entire human genome, to read out all the A's, G's, C's, and T's in an individual person. It took years and years. We now have automatic gene sequencers that work like computers. This is a gene sequencer, where basically you put the sample of DNA and it reads it very, very quickly. So we can do that much easier. We can even create our own DNA. You can make synthetic DNA, because DNA is really just nucleotides, right? Combined one after the other. And scientists now can synthetically take an A, bond it to a T, bond it to a G, and create their own genes, which can then produce various types of proteins. So this is a huge growing area, and anybody interested in biology and these things, this is an area probably that is a field in which there will be a lot of work and education and jobs, because it's such a new field. These pictures basically are just showing a couple things. This is showing when this DNA molecule gets cut by a restriction enzyme. Again, it cuts it between certain base pairs, and it forms what we call these sticky ends. And they're complementary to each other, so when they're put back together, they sort of stick back together like Velcro. So if you cut the bacteria plasmid with one restriction enzyme to open it up, and then you cut the DNA molecule with the same one, when you put it back together, it all sort of seals back up because of these sticky ends, which are complementary to each other. This is a DNA sequencer, and this again is just showing how that works by sequencing a long strand of DNA. This is sort of again how you would take and splice a gene from one organism to another. This talks about growth hormone rather than insulin, but the process is the same. You start with a human cell and a bacteria cell. You find the gene you're interested in. In this case, it's a gene for growth hormone. Okay? And you cut it. You cut that area of the DNA out using a restriction enzyme. So here's the piece. This is the gene that tells our body how to produce growth hormone. At the same time, you take the plasmid out of the bacteria. You cut it with the same restriction enzyme to open it up. You then combine your gene that you're interested in with the bacteria plasmid. Because they were cut with the same restriction enzyme, they have those sticky ends that are complementary to each other. And what you end up with is a combined plasmid, which has the human growth hormone gene in there. Put it back into a bacteria, culture the bacteria, and then we can produce human growth hormone in a bacteria. So, some more examples. Transgenic, that term just means it's an organism that contains DNA from another species. So, some examples we've talked about: insulin, growth hormone, clotting factors that are produced by bacteria. Transgenic animals. Mice have been genetically engineered to have cancer genes in them turned on. So these mice get cancer.
Why?
Good question.
So they can try to find a cure.
Yeah, they use these mice in researching cancer treatments. So obviously, you're going to test a new form of chemotherapy. You need to test it on an animal first. And so they use these mice to test it on to see how they affect the tumors in the mice. Livestock that produce excess growth hormone inside of their cells to grow larger, to be more productive, to produce more meat. Animals that produce medicines in their milk. Transgenic plants, very common, and sometimes controversial. Plants that have genes that produce insecticides, like we talked about. Plants that are resistant to Roundup Ready. Golden rice that produces extra vitamin A to give in areas where there's a deficiency. One of the controversies is that companies that create these transgenic crops, they're copyrighted. In fact, if you grow Roundup Ready corn, you're not allowed to save some of the corn for seeds. You have to buy the seeds from the company that produced it. These organisms are actually copyrighted and are, because a company produced this organism by doing their genetic engineering, and so you're not allowed. If you ever watched the documentary Food Inc., they talk a lot about Monsanto and trying to, they copyrighted these seeds and you can't grow these crops for seeds. Lots of times they're also engineered to be infertile, so that you can't even if you did gather them, you couldn't grow them. You have to buy new seeds from the company every time you're going to grow these crops. Cloning. You heard of cloning? Cloning is a form of new genetic technology. The first clone was a clone is just a genetic copy, basically an identical twin of an organism. And the first cloned mammal was produced in 1997. It was a sheep. You know the sheep's name? Dolly. You guys still know that?
Yeah.
How do you know that? I did? Okay. Yeah, Dolly was the cloned sheep, the first cloned sheep. Since then, many other mammals have been cloned. No humans have been cloned to our knowledge, although the process would be the same. People claim to have cloned a human, although they never provided DNA to prove it, so it's probably a sham. But in cloning, what happens is it's not all that complicated really, but what you do is you take the animal that you want to clone, in case this sheep, and you remove the nucleus from one of its cells. It could be any cell really. Any cell from that sheep. In this case, they used a cell from the udder. Basically, sucked out the DNA using microscopic dissection tools. Took out the nucleus from this. Then they have to take an egg cell from another animal. Okay. and basically they take this egg cell and they get rid of the nucleus, just throw it away. So you have an empty egg cell, okay? And then you have the nucleus of this species we're trying to clone. Basically, you just stick the nucleus from the one you want to clone into this egg cell. Okay? Give them a little jolt of electricity, and that sort of stimulates that egg cell with the new nucleus in it to start to divide. And it acts much like a zygote. It divides into an embryo and grows into a fetus, but all the chromosomes in that cell came from one single individual. It will be genetically, this sheep, then they have to implant that embryo into host mother, into the uterus of another sheep so it can grow placenta and she can give birth. But when she gives birth to that sheep, it has none of her DNA in it. It's a clone sheep. It's identical to the original. I believe you can pay to have like a patent clone. You could send a sample of DNA from your favorite cat, and I believe they will clone it.
What if you're like,
Wait,
is Dolly still alive?
Dolly's dead now. Dolly's dead.
You could have decided to
I don't. So often, some people say that the clones when they do this cloning, often many of the embryos never survive, and some that survive as they age don't age normally. There's more complications, more health effects with the clones. So yeah, there are some consequences of that. Is that the last?
Yeah.
All right, let's watch this. This is kind of cool. We'll be done.
You know how much it is to clone your dog?
I'll look it up when you guys are working in your lab.
Probably like
It's expensive, I'm sure.